As the world faces a new era of global climate change and biodiversity loss, environmental scientists have
It was working for a second.
been unable to keep up with the global challenge of surveying and documenting our changing ecosystems. Proper biodiversity surveys are critical to the creation of conservation plans and to developing our understanding of how humans are impacting the Earth. However, traditional methods of field sampling, which require large teams of well-trained scientists, are few and far between. If we hope to address the low amount of high-quality biodiversity assessments around the globe, scientists and policymakers alike must embrace remote monitoring techniques. Now, let's travel to Tetiaroa Atoll, a resort island in French Polynesia once treasured as a vacation destination for royalty and known for its high biodiversity. After visiting to film Mutiny on the Bounty, actor Marlon Brando became passionate to protect its cultural and ecological beauty. However, this idyllic island is plagued by invasive rats, which prey on native sea turtles and seabird species. Island Conservation and the Tetiaroa Society are working to remove invasive rats from the island, which has great benefits for the terrestrial ecosystem. This work may also enhance seabird-driven nutrient cycling to the ocean, improving the adjacent coral ecosystem offshore. Extensive efforts are being taken to generate and standardize comprehensive data for Tetiaroa Atoll. However, many current monitoring methods have been through traditional surveys, requiring researchers to travel and encroach on remote areas to collect biodiversity data. This process is time-consuming, leaves data gaps due to sampling frequency, and can be prohibitively expensive. There is also an added risk of potentially carrying invasive species onto the island. However, innovative monitoring techniques like eDNA can make these efforts less strenuous. Environmental DNA is DNA collected from environmental samples such as soil, seawater, or air. Unbelievably, through laboratory techniques, researchers can identify the species present in a location using eDNA. eDNA also has the potential to address many of the issues of traditional sampling, as its fieldwork can be done by anyone. eDNA community science programs are popping up all over the world, and comparative studies of eDNA versus traditional monitoring are underway. Our team is BlueWaltzBio. As you saw, we showed you our video. So just as a quick timeline of what we're going to talk about, we're going to talk a little bit about who we are, how we left our original problem statement and then pursued a variety of different fields, how we found our very first pain point in eDNA, and how that basically led us on a journey to pursue that field, find other pain points, and develop an MVP in a wireframe, and where we ended up.
Okay.
So this is our team. Just really quickly, we're a group of 3 undergraduates and 1 graduate student. Our sponsor was Island Conservation Heath Packard, and we presented one of their problems in the video we just showed. And Craig is our mentor, and he's a great guy. So as we go through this, we're going to explain a little bit about how we move forward using this visual. So in the early weeks, we were a little lost. We were searching for a problem, and we kind of felt like we were metaphorically lost in fog. So the initial problem statement we were given was to create an autonomous eDNA robot, because in theory, it could solve the issues of field sampling, provide continuous monitoring data, and it could potentially be cheaper as well. We did a little bit of further study into this and found that organizations with tons of resources like MBARI and other major organizations were already working on this, and we deemed it infeasible for us to pursue. In hindsight, we believe that this might have been a mistake, because we shouldn't, as some teaching staff pointed out to us, we shouldn't pivot because we don't feel like we have the skills to approach a problem. We should pursue things and then add people to our teams later if we find a valid issue. But regardless, we left this issue and set out into the fog, so to speak. So in our early interviews, we talked with people in a variety of different fields, and we really just weren't sure where to go. Essentially, it felt like we were kind of just reaching out and talking with different people, hoping to bump into something. And then in week 3, we actually did, which was incredibly exciting. So it all started with Abigail Wells, who is a molecular geneticist at NOAA Fisheries. She was our very first eDNA interview, and the very first scientist we spoke to that does that work on the ground, as opposed to managerially. So one of the key insights she pointed out to us is that usually in labs, one person that is the eDNA specialist is required to do field work, wet lab work, and bioinformatics. This can cause various problems because people are trained in different fields. So one issue she presented to us was that pulling sequence data from GenBank and the NCBI website can take her days to weeks, depending on how big of a data set she's trying to pull when she's starting a project. And she said to us, I would thank God if I could download the list of sequences all at once. This problem makes a lot of sense because she's trained traditionally in wet lab and field work and doesn't have very much programming experience. So we were overjoyed to have a problem, and we decided that we were going to go towards eDNA in the fog, and we left behind the other topics of satellite remote monitoring, water quality, et cetera, not because we didn't think that they necessarily didn't have problems that we could find in them, but just because we had found something and we really wanted to pursue it.
So
we had a working hypothesis at this point, which was that eDNA scientists are largely traditionally trained ecologists and thus don't have programming experience to automate their bioinformatics work. And this ends up with them wasting a lot of their time. So we had heard this problem, but we didn't know a lot about the eDNA space in general. You can go to the next slide. So we spent the next couple of weeks diving into the eDNA space and understanding what people do once they get that sequence data and what the next steps would be. So we went into interview overdrive.
So we completed 26 interviews in 2 weeks. And we found the interviews by reading an article and then like contacting all the authors. And so we created this graphic which explained the whole process. And then we tried to, every time we interviewed someone, we would ask them what their greatest pain points are. And we found the pain points, a lot of pain points were in validating primers or finding a genetic barcode that is unique to one species. Because part of the job that Abby and other environmental DNA technicians really hated was finding a unique DNA barcode that identifies just the species you want and nothing more. And part of the reason that they had so many problems is because of the database that they were using while they were doing this, GenBank. When they're using GenBank, it's like sort of it's created by the public and anyone can add stuff to it. And it's very unstandardized. So it's incredibly painful if you're trying to find a single like location in the DNA to see if that's a good genetic marker, barcode. It could be called 3 different things. It could be called CO1, COE, or the full name chrome oxidase 1, which makes it very hard to search. The location could be in multiple different fields, like in the title instead of in the proper like location metadata field. And the species name could change, which would mean that you wouldn't get the you wouldn't get your species list. It wouldn't be able to download everything you were interested in. So we created this chart, this product market fit chart, to see their value. I forget what it's called, value proposition, and that we would make one integrated website to download, filter, and clean data for ecological geneticists. And so we quantified the problem using a few quotes. From Michael Young, we figured out the number of labs, where there's about 100 to 200 labs, and it's hours and hours of highly skilled laborers' time, an estimated of over $600,000 interacting with this time spent interacting with this database. But then we hit a bumpy road when we started testing our MVP in wireframe. So here's our MVP. Here's the search page with the key bit is the file upload, which is the batch downloading of everything. Here's the summary page that you could then export to your other softwares. And so what we found is we did another 20 in our 21 interviews of alpha testing is the best feature is the batch downloading. And we also found a bunch of new features to help deal with the nightmare of mixing databases to try and use like other data sources, other databases to take into account name changes that might have occurred and not been put in, and the GenBank thing might not have been updated. And also to see the coverage of your species list inside the database to see if the database contains every species you're interested in. But then the problem was we were competing with bioinformatics specialists in almost every lab, it seemed like. Once a lab got to a certain size, they would have a bioinformatics specialist who could make an R pipeline to pull and clean the data. And so we created an open source hypothesis that we were going to make a graphical user interface for all the open source used in primer development. And we would expand into making this GUI for a bunch of different software that people had to either write themselves, which took them a long time because they weren't trained coders, or like, yeah, that each lab was doing itself. We would make a nice GUI that could be used. But then we found out that their Genius exists, and they've already done that. They've already created a graphical user interface wrapper for all these different things, for all these different open source softwares. So some of our teammates, Sam and Ray, went undercover to try and figure out more about our competitor, what capabilities it had, and to get a free trial. And in our learnings there, we found out that we were really only one feature, like at least in our like long-term plan, we were really only one feature ahead of Genius. Only a few features that could be easily copied. The batch download feature could be, once they figured out that that was a real big pain point by looking at us, they could easily copy it and then take away our whole market. So yeah, that was some of the industry knowledge we created. And we also did a bunch of research into how they bought software in academia and government, and found that there's a ridiculous amount of friction. You have to go to your supervisor, you have to fill out a bunch of papers to prove that you actually need it. It's much more difficult than it would be if you want a piece of software and just sit down and buy it, you know, just one click. And there's a lack of funding, and then there's competition with writing the code yourself and with the open source community. So we're competing with free people, which means that it's like, it felt sort of like we're offering a water bottle inside of a store instead of, you know, in the desert, as you guys kept on bringing up. So in conclusion, we found that we had not found a product market fit, which was really a bummer for us because we wanted to create a profitable company when we set out here. But what we did find is a problem solution fit, because we did have early adopters who said they would use it because of how painful our code is. Even the best coders we found often said they would still use this because of how annoying it is using the code they've written. But we failed in getting users to pay. So some of the major lessons we learned is don't pivot based on your current team's skill set. Because at least in this class context, the process was just trying to get the problem solution fit, not really about creating a, trying to find that market that you could fill, that niche. And really understanding the difference between the product market fit and problem solution fit really hit home. And I think the third big key takeaway is we needed to spend more time brainstorming novel solutions, because it seems like we would take like the first solution that came up for the many problems that we had, and then we would just run with it. And we didn't spend very much time thinking like, how can we like come up with something really new and original, which is why when we found Genius existed, you know, like we were screwed. Because they already had done it. So moving forward, we need to explore alternative fundings, because there is still a market for making these primers that are specific to the species you want. There doesn't seem to be any like tools that do the whole thing. So see if there's any grants or outside sponsors. We need to explore the feasibility of the batch download, and by talking to GenBank and ensuring that partnership can work. And we need to see if there's any other major pain points we can integrate, like data visualization or finding database coverage, which GenBank doesn't have and wouldn't be easy to copy, so that we can successfully make it and become a big enough pain that people would be willing to pay, or funders would be willing to fund us. And yeah, thank you very much to Craig and Heath and the teaching team. This has been like a real blast for me through all the ups and downs and a lot of stress, but a lot of good time too.